All right, well, thank you first to Alex's Lemonade Stand for hosting these seminars in this unreal time that we're all living through. And thanks for, really remarkable audience for the first lecture I've ever given from my home. It's a privilege to have been Alex's doctor and to have worked with the foundation through all these years. And I think everyone in this call is aware of the impact that the foundation has made on childhood cancer over the year and how they've adapted to this pandemic and how they're helping families in addition to all the research they support. So thank you very much. So I'm going to talk about precision immuno-oncology, and really the reason why I say it starts with optimal targets is I'll try to talk you through is that childhood cancer immuno-oncology is very different from immuno-oncology for adults, and we need to think differently about it, and I'll try to convince you of that as we go through. So over the next half hour, what I'd like to do is just talk about the rationale and the infrastructure that we've built over the last few years for a coordinated pediatric cancer-specific immuno-oncology effort. And then I'm going to give you three examples of things that we weren't thinking about many years ago that are now turning into clinical opportunities, and hopefully one, if not more of these, could be potential effective precision therapies for children. These are all neuroblastoma focused, but as I'll try to emphasize that while these were discovered in neuroblastoma, we're learning that many of these targets are more broadly applicable to other childhood cancer diseases. Then I'll briefly end with a more general discussion of the landscape and where we're heading in pediatric cancer immuno-oncology, and sorry for the misspelling, in our future directions. So I think that everyone knows that, for those of us who've been in the game for a while, throughout most of our careers, there were no new FDA-approved therapies of any type, very few in childhood cancer. And over the last half a decade, we're witnessing several new effective therapies being translated into FDA-approved medicines, and all of these are immuno-oncology agents. So the dinutuximab, which showed an improvement in event-free survival for children with neuroblastoma in a phase 3 trial, completed over a decade ago now. And then, of course, CAR T cells really changing completely the landscape of first relapsed high-risk ALL, and now making an impact likely in newly diagnosed ALL. So these are great examples. But as I mentioned in my brief introduction, kids' cancers and adult cancers are very different. And maybe this list is obvious, but let me just go through it. So, of course, childhood cancers, children who develop cancer, they're innocent. The environment has little and sometimes no influence on tumor initiation, whereas adult cancers have a major environmental influence. That correlates with the fact that childhood cancers generally have very low mutation burdens. It's not the sole reason why, but low mutation burdens, whereas that's the opposite in many adult cancers, not all. As already mentioned, there are a few new FDA-approved immunotherapies for kids, and in adults, there are hundreds of recently approved FDA therapies, but these are generally checkpoint inhibitors in a variety of different indications. So, what I've noticed over the years, and maybe many of you as well, if you walk into a medical oncologist's office or meeting and ask what immunotherapy is, it's checkpoint. And whereas if you were to walk into a pediatric oncologist office and ask, it's CAR T cells and maybe antibody drug conjugates. So we're thinking very differently about things. Of course, the host immune system is radically different in an infant compared to an octogenarian. And what we've learned is that a lot of times what we're doing is trying to figure out what on the surface of a cancer cell, whether it's in the plasma membrane or presented on MHC, what we can target. And in many cases, the adult cancers are reflecting the tissue of origin, whereas in the childhood cancers, it's generally developmental proteins that give us some advantage. And I think it's this difference, the developmental origins of childhood cancer, that really is why we're focused on a discovery effort. In both cases, however, there's not nearly enough validated targets that are truly tumor specific, and that's a big effort for many people around the world trying to think about immunotherapy for cancer. So whenever I show this slide, I feel like I'm cheating on Alex's lemonade stand, because a different foundation helped us with the Stand Up to Cancer Foundation to get this project off the ground back in 2013. And this was a coordinated effort of the centers that you see here, and co-led by Crystal Makel and myself, to really think differently and use the power of genomics to discover and develop immunotherapy targets. And while the initial round of funding ended in 2017, we felt that with our Patrick Sullivan and a large group of advocates, and we had over 100 investigators working on this project, that we didn't want to end after 4 years. And we've been able to leverage our progress, we think, in the first 4 years to successfully translate all of the early discovery work that we're doing in several different moonshot projects, NCI-funded moonshot projects. We do continue with St. Baldrick's funding, as well as our institutions matching funding. And then also through the moonshot was money appropriated for the first pediatric cancer-specific immunotherapy clinical trials network, which is just launched this year and is led by Crystal Makel and Julie Park, and the institutions you see here. So this is the infrastructure we've built, and has led to, we think, a lot of exciting work, only a small bit of which I'm going to discuss today, but hopefully we can get some other investigators from this network to talk about other projects in this ALSF lecture series. So I want to tell you about three targets that were discovered through this effort, and how we're thinking about them and what their status of development was. So one of the very first targets discovered through this effort was Chris Bossi's work at Children's Hospital of Philadelphia, in collaboration with many people throughout our consortium. And I'm not going to go through what was published back in 2017 now, but what Chris did was use a very large neuroblastoma specific RNA sequencing and whole exome, whole genome sequencing data set that we had generated to to develop algorithms to define proteins, mRNA that encode proteins that are highly overexpressed homogeneously in neuroblastoma tissues, and then absent or very low expressed in normal childhood tissues. And so GPC2 met those criteria. But we also felt it very important for the proteins that we're going to prioritize for immunotherapy development to be essential to the sustenance of the cancer, so that these are a function as an oncoprotein. And so Chris showed in this paper that if you deplete GPC2 genetically or therapeutically, as I'll get into, neuroblastoma cells don't tolerate it. And that he defined the mechanism of this is shown in this cartoon, that it is a direct MYC-N oncogene target gene, so MYC-N drives expression. And then in the neuroblastomas do not harbor MYC-N amplification, the gene sits on chromosome 7q, and gain of that locus leads to overexpression, and that's a very frequent genomic event. So that's how he used genomics to get at GPC2, and then worked with our consortium and Mitko, Dmitry Dimitrov, who's now in Pittsburgh, to develop a highly tumor-specific binder, or an SCFV, that we could be built into reagents to use as immunotherapies. And so the unpublished work from Chris, part of it is shown here, and just shows how he has taken this forward and now hopefully nearing translation to the clinic. And that has, of course, been slowed a little bit by our not being able to be in the lab, but he's moved this forward very quickly. So this is a crystal structure of the extracellular domain of GPC2, and this is the D3 binder built into an FAB shown interacting. And I'm not going to show all the data here, but this is interacting not in a linear portion of the protein, but in a conformational. So it's touching on multiple different helices. And so you cannot, like in the CD19 experience, where one mechanism of escape is to delete the 2 exons that mediate binding of the CD19 directed CAR, that can't happen in this situation. The other thing that Chris showed that I think is very elegant is that tumors express the full length protein, exons 1 through 10 are expressed, whereas in normal tissues where it is expressed at low levels, it's only exons 5 through 10 through a cryptic start site in intron 4, and that this is touching on epitopes that are, or protein junctions that arise from the early exons as well as the late exons that suggest significant tumor specificity. He also showed in this membrane protein array, this is just taking the binder and seeing if it happens to stick to any other, so this is an array of every single known membrane protein. The only thing that it binds to is GPC2, which is suggesting there's not going to be some off-target effect. And then he's shown in these patient-derived xenograft models of neuroblastoma, he's done about a dozen of these, that relatively low doses of this antibody attached to a PBD, it's an intercalating agent used in several antibody drug conjugate designs, can regress tumors and cure mice with relatively low doses, single dose or multiple doses. And as I'll show you in a minute, this protein happens to be expressed in other cancers, including small cell lung cancer. And we did these experiments hoping to get industry interested even more than just the childhood cancers. We think that this is a good target. And again, this is showing potent activity at relatively low doses of this PBD dimer. So one of the themes of this talk that is that we are big believers because of the lack of therapies for many children with high-risk cancers, especially relapse cancers, that we need multiple shots on goal so we don't put all our eggs in one basket. Anyone who does drug development on this call knows that, you know, things can fail at any step of the way. And I'll come back to that point in a second. So Sabine Hetzinger in Crystal Makel's lab has been spending a significant amount of time building different CAR constructs. This single slide does not do justice to the amount of work that she's done to, you know, make multiple different types of constructs to find the optimal one to bring forward. And so this is showing data with CAR constructs that are ineffective, and then CAR constructs that are effective. And there's a little bit of a difference depending upon whether or not the co-stimulatory domain is 4-1BB or CD28. But you can see that these mice have outstanding, this this CAR is potently active in both of these configurations with the 4-1BB, consistent with what we know from the CD19 experience, perhaps persisting longer, but the CD28 perhaps being a little bit more potently active early after infusion. And so as we move this forward with an interest in neuroblastoma, Chris has really done a nice job at sort of mapping out what other cancers may be of interest. So this is a kind of a tricky slide to read. This is showing on this axis in blue, the percentage of tumors studied. So 247 neuroblastomas studied from 4 different data sets that about 78% of them have an expression level that we think is high enough to be targeted with immunotherapy. In that, in red is shown the median expression, so that if you express it, there's a tight range, and it's generally highly expressed. And you can just look across this in your favorite tumors, and we use these data to think about how we might be able to design future clinical trials where the eligibility criterion would be likely an immunohistochemical assay with a staining pattern above some certain H score, a certain degree of staining. And so those are in development as well. And while we'll likely start in neuroblastoma, we certainly hope to expand this to other histologies. So where this stands presently is that we've had a hard time with the ADC manufacturing. As good as that looks, we've worked with a couple of companies. Every company has their favorite payload and design, and just to be quite honest with you, that's been frustrating. We have not been able to move that forward, and we're thinking about ways to do that in-house, and that's a little bit tricky to make GMP antibody drug conjugate in academia. But the CAR stuff is moving forward very quickly, and there are 2 CAR T cell companies who are heavily invested in this and moving things forward. And again, this is not a competition, this is a collaboration, because as we learned with CD19, we learn from different types of CAR constructs going to the clinic what works and what a liability might be. And then finally, Chris and Guillaume and others in the PIDDN are working on bispecific approaches, both T-cell engagers, as well as trying to increase specificity by going after GPC2 and maybe GD2 or maybe NCAN. And that work is in the early days. So I'm going to move now to work done by Amber Weiner, who's primarily in Sharon Diskin's lab. I helped co-mentor her, but Sharon, who actually has a ALSF Innovation Award to kick off this work. And what Amber, who is a graduate student, her dissertation was really about, yes, Chris showed that you can look at RNA-seq data, and in his case, the RNA-seq data was highly predictive of the protein expression. But a lot of time that doesn't happen. In fact, probably about 50% of the time. So Amber developed a method that I'm not going to get into in detail here to purify the plasma membrane away from, she started with neuroblastoma cell lines, then she went to patient-derived xenografts, and then primary tumors, and to get it away from the rest of the proteome, and then do mass spectrometry, and then to integrate that with our epigenomic, genomic and transcriptomic data. And so what she showed is that when you do the proteomics, you find things that we know, which is very reassuring. So as you know, there's a Julie Park, Mike Jensen have a CAR T program in Seattle focused on L1-CAM. We talked about GPC2, so these are some other card-carrying members that we know are overexpressed. But what popped out that we had not seen before is a protein called DLK1. And what was different about DLK1 is that, and you can kind of see it in these data here showing blue is abundant, white is not abundant, that unlike NCAM that is uniformly overexpressed, DLK1 either seemed to be on or off. It's not quite that binary, but some neuroblastomas expressed extremely highly and some didn't express it at all. And then what Amber and Sharon were able to show is that we searched for the mechanism of that. And is it a MYC target gene like GPC2? The answer is no. Is it a locus that is amplified? The answer is no. But Amber turned to our H3K27 acetyl data, which, as you know, marks enhancer elements, or when they're really stitched together, super enhancer elements, and showed that if you look at these swoosh plots, which I think many of you are familiar with, which is showing the ranking of super enhancers in each of these models, things like MYC will be up at the top here because it's amplified. Other things like FOX2B and other very important oncoproteins are near the top. But DLK1 was very highly ranked in several of these, but then way down at the bottom in several others. And that correlated completely with expression. So if you had an enhancer, you had DLK1 expression, and if you did not have an enhancer, you had no DLK1 expression. And this is just the pile-up data of the H3K27 data. So we had a mechanism, and as you know, most of these super enhancers in cancer seem to be locked in, and we think that if you have this super enhancer, this overexpression is not reversible. That's something we're still working on proving, but that's the hypothesis at least. Now, it just so happened that a company called ADC, Antibody Drug Conjugate Therapeutics, had an antibody drug conjugate, sorry about that, shown here. In development, DLK1 happens to be overexpressed in hepatocellular carcinoma, and they are launching a trial in Asia for patients with hepatocellular carcinoma. But we were able to get our hands on this reagent in collaboration with them, of course, in the Pediatric Preclinical Testing Consortium. And these are the xenografts that we studied, and you can see the staining here, and it's a little bit difficult to see at this magnification, but this is more purple than brown, meaning negative. This is a lot more brown than purple, meaning positive. This is sort of ordered in the gradation of absent, low, a little bit higher. And then you can see that this single dose, 1 mg per kg, show no activity when no protein's there, a little bit of activity when a little bit of protein's there, but then, very potent activity when it's moderate or high expressed. This is one tumor that escaped and then responded to a second challenge, again, confirming that DLK1 expression is not lost as a mechanism of resistance. So like GPC2, this was discovered in neuroblastoma, but this is extremely highly expressed. So here's neuroblastoma. RNA expression, high, medium, but there's a wide range because it's either low expressed or high expressed. This does have a liability in that it's normally expressed in the adrenal gland. So it is highly expressed in the adrenal cortex, and it is in some other normal tissues that may be not as essential. But then not surprisingly, this is very highly expressed in adrenal cortical carcinoma, as well as pheochromocytoma. So what we're planning on doing is, in collaboration with the NIH Rare Tumor Center and Brigitte Wiedemann, is to launch a trial that will first be in adults, primarily ACC patients, but maybe pheo patients as well, for dose finding and safety. And if that is found to be safe, we would rapidly expand within the same protocol to children with neuroblastoma. And as I discussed with GPC2, Wilms tumor seems to be another good target. There's a subset of AMLs that very highly express it. So whether we would just do neuroblastoma or go to a basket trial still needs to be determined. So the last story I'm going to end with is work that Mark Yarmarkovich did during his dissertation in his lab. He is now a postdoc in my lab. And Mark asked the question, despite the long-term knowledge that neuroblastomas are very low in MHC class 1 expression, have very few T cells in the tumor microenvironment. They're the classic, in fact, maybe the card-carrying definition of what a cold tumor is. Could you target peptides presented on MHC in neuroblastoma as a proof of concept that if you could do it in neuroblastoma, then you could probably do this in most human cancers. And so what Mark did was a sort of a twist on Amber's work in that he had all of our genomic data, which you can do a lot with in terms of inferring HLA type and differential expression of genes and that sort of thing. But what he did is he developed the technology to pull down the MHC class 1 and class 2 using an antibody, and then to elute the peptides off of that for mass spectrometry. So purifying from tumor tissues, he started with PDXs and then primary tumors to get the peptides presented on MHC specifically eluted out of these tissues, and then to quantify those to look for tumor-specific antigens. And again, I'm not doing justice to a huge amount of work that he did, but this is one way to look at his data. And so shown on the top of this plot in red are the peptides that he eluted from neuroblastoma tissues. So there were about 11,500 peptides that he eluted from 5,000 genes in this one experiment. And then also we had available to us, our collaborators in Tübingen, Germany, they've done this exact same experiment on 92 normal tissues. And so, you know, this is these are a couple of peptides that are very highly expressed in all of the neuroblastomas. So 100% of the neuroblastomas had these peptides, but they are also on some normal tissues. So these were excluded. And so what we're really interested in are the peptides over here, 4,000 or so that come from 2,800 genes that where it's really neuroblastoma specific. And really what we found, at least I found incredibly impressive, is that if you take these 2,800 genes and just do a gene enrichment analysis and ask what terms hit, the only term that hits is sympathetic nervous system development. And so we obviously want to concentrate on the peptides that are recurrent. And so we developed a prioritized list. And for those of you who think about neuroblastoma, you'll recognize many of the genes where these proteins came from, that these are, you know, key oncoproteins, key regulators. These are generally overexpressed transcription factors that we don't know how to target. And so we prioritize those that are were found in tissues where there were a common HLA allele. It was highly abundant, and they bound with very high affinity. So we would look for things that bound high affinity to us, anything under 500 nanomolar. So these are things that were, these were peptides that were prioritized. It's probably obvious from what I said, but what Mark did not find, which is not a surprise, and is again highlights one of the major differences between childhood cancers and adult cancers, is he did not find zero neoantigens. So these are all wild type peptides that are found recurrently. And so unlike a mutant KRAS that may get on HLA, and you have your you have a sort of an antigen that the immune system has not seen before and gives you some specificity, we think that these are good self-targets because in general, these proteins are turned off in childhood tissues and adult tissues, giving some specificity, which is part of the filtering criteria. And so working with, again, Dmitry Dimitrov, Mitko, Mark developed a very clever system to screen for SCFEs by first putting a decoy peptide on these MHC tetramers and depleting a phage display library with things that would bind to the HLA and not the peptide in the HLA, and then to do 3 rounds of enrichment with the specific peptide. And he got things like this. So these are ELISAs, and this has a little bit of specificity, but we were really looking for things like this that were no binding to the decoy and specific binding. And this F11 is really probably the best one we have. And so Mark has built both TCRs and CAR T cells, and here I'm showing the CAR T approach, and has shown that he can take neuroblastoma cells in a dish, put in these CAR T cells with the design that you see up here. And in this is caspase assay, you can see that the specific CAR construct shows potent killing red in this time-lapsed effector to target ratio 2:1, whereas these cells become confluent and all this red is just background red, and there's really no killing by this non-binding CAR T construct. So this earlier days, Mark had geared up and made all the T cells to do this in mice when we needed to shut down the lab. So it's a little bit further away from the clinic, but we think the concept of targeting peptide MHC complexes in childhood cancers is a valid approach, something that we would like to pursue. We think that we can get around the HLA restriction by finding peptides on a variety of different common HLA alleles. And we've just begun a collaboration with Mike Taylor and his group in Toronto to apply this work to medulloblastoma as well. So just in summary, last 2 slides. When we started our efforts, Crystal and I and our team, these were the known immunotherapy targets. And I've talked about a couple others, but these, and again, we don't mean to take credit for all of these, but many of these are a large focus of our group, but this is the current landscape of what people think might be good pediatric cancer specific immunotherapy targets in 2020. And across our consortium, there are a lot of collaborative efforts going after several of these. And as I think everyone on this call knows, one of the huge Achilles heels in the solid tumor space is tumor cell heterogeneity. And it's very likely that many of these, well, some of them already have, and Crystal and Michael and several other people in the group just had some two beautiful papers in Nature Medicine, and one of them showing the potential for multi-target targeting or multi-specific CAR T cells in pediatric brain tumors. So 2 broad conclusions. In neuroblastoma, we think that a lot of the oncogenic drivers are non-mutated oncoproteins that are master regulators of the sympathetic nervous system development, and thus give us an advantage that they are not normally expressed in childhood or adult tissues. And we're developing several of these, as I showed you, as immunotherapeutic targets. And that, this applies not just to neuroblastoma, but to all of the developmental malignancies. And, one of the big things we're trying to do through the PED-CITN is to develop very well validated biomarkers to select patients for these trials based upon the expression of the target protein of interest. So this is, huge work, but lots of people within our consortium, my lab, who they're great in the way that they continue to be positive and working together in these unreal times is very much appreciated. Alex's and other funding agencies, collaborators at CHOP and Penn, and many others across the country. So, and then I mentioned Chris and Mark and Amber as we went through work I'm presenting here today. So thank you for your attention. Hopefully people are still there.
Thank you very much, John. I apologize that our internet went down when I was doing your intro earlier, but you did a great job. I want to thank you so much for the talk. Just to finish up on your intro, John has been a big help to Alex's Lemonade over the years. He's been a founding member of our scientific advisory board, helped us develop our path for funding research, new areas to get into. And on top of that, he was also Alex's doctor and the reason we moved to Philadelphia. So thank you for that talk, John. And I'm going to jump right into questions. So if you have questions, you can send them via chat and we'll get them to John and he'll answer them. So the first one is from Martine Roussel. Can you comment on the high expression of GPC2 in testes and possible effect of CAR T GPC2?
Hi, Martine. Thanks for that question. So what we think, and we're trying to prove at the protein level, is that it's a different isoform. It's a truncated isoform, and we haven't been able to prove this yet, but the hypothesis is that the SCFE that we've prioritized will not bind to testes expressed GPC2. Can we prove that there isn't some full-length expression? We have not done that yet, but it is something that we'll absolutely have to pay attention to as this moves forward.
Thank you. The next question comes from Quinlan Marshall. And it says, you mentioned the development of antibody drug conjugates against DLK1. Is the development of a CAR targeting DLK1 possible given its high expression in normal adrenal gland tissue?
So Quinlan, it's a great question. And there we've decided to steer away from CARs because the adrenal expression is so high. And why we think that we might have a therapeutic index with the antibody approach is that we think internalization kinetics are going to be different in cancer tissues versus mature adrenal cortex. We haven't proven that yet, and the real test will be when we get into the clinic, but we're just concerned that a CAR T might just be, persistent. If we can get a persistent CAR T, it would just be too toxic due to the normal adrenal and also pituitary expression.
Okay, thanks, John. You have a couple people who said great talk. And then another question came in. What do you think of MIBG therapy?
Well, that's an interesting question. We have spent a lot of time over the years working on MIBG therapy. It was actually the therapy that Alex came to Philadelphia to receive back in the days when it was highly experimental. It is active therapy. Actually, what it binds to, the NET receptor, we actually think is also a good immunotherapeutic target, something we've been thinking about. And it's being studied in the current phase 3 trial to determine whether or not it can increase cure rate. So it is the oldest targeted therapy that we have for neuroblastoma.
Okay, next one comes from Hassan Uladag, who says, thank you for the nice overview. Can you articulate why producing ADCs have been so difficult to produce industrially?
Yeah, we've had 2 collaborations, and the bottom line for us was that the toxins that they wanted to work with are good for adult cancer. So these are generally the metastanoids, and they just are not active in neuroblastoma. And so, they were not willing to use. So Chris has done a beautiful experiment where he's looked across a large panel of neuroblastoma cell lines with all of the different toxins that have been built into ADCs. And generally, the intercalating agents, any of the chelators are very active across neuroblastomas, where any of the more traditional poisons that are in, for example, trastuzumab are not active in neuroblastoma. So, that's been our handicap. Chris has a new paper out, and he's just reminding my text that his work was funded by Alex's NIH Stan Foundation as well. I think that with this new paper, we're hoping to get more industry buy-in. So we haven't given up, that's for sure.
Okay, thank you, John. John, we have a couple of people that are raising their hand, and I think that means they want to ask a question with voice, but I'll do a couple more chat ones, and then maybe we'll go to them. Sure. Okay. So the next question comes from Manaj Basin at CHOA and Emory in Atlanta. Can you comment on splicing-based neoantigen in neuroblastoma?
Yeah, so I mentioned briefly the project with medulloblastoma, and as you may be aware, Michael Taylor and his group published this beautiful paper in Nature a few months ago about the most common mutation in medulloblastoma, and about half of the sonic hedgehogs is in the U1 splicing machinery. And they show very nicely in that paper that leads to novel splice isoforms that drive medulloblastoma initiation and progression. So that's exactly what we're doing, is we are looking for those novel splice neoantigens in that situation where we have both U1 mutated and U1 non-mutated samples from his lab. So be very excited for Mark so, it was another casualty of COVID that, right before we had to close down the lab, we got all of the RNA sequencing and samples from Michael's lab, and the plan is to do the mass spectrometry that MHC pull-down mass spec that I showed on neuroblastoma on those samples. So stay tuned.
All right, John. Well, Carolyn Felix has a question, and she has her hand raised, so I'm going to try to allow her to ask the question. I haven't done this before, but we'll see how it works, okay?
Sure.
All right.
You there, Carolyn?
I got to unmute her. I'm having trouble unmuting.
I mean, I see your question, Jay. It says,
yeah.
Are the relevant enhancer sequences that you mentioned mutated?
Yeah. Okay.
And so, Carolyn, to the best of our knowledge, the answer is no. But as you know as well as me, that's not a trivial thing to exclude completely. So we just got in a huge new batch of high quality whole genome sequencing data. And Alex Lee in the lab, a graduate student, is actually doing just this to look at potential mutations within super enhancer elements in neuroblastoma. So I know where you live. I can let you know when we have that result.
All right, thank you, John. I'm not going to try to do another raised hand, since that one I couldn't unmute the person, but somebody else said interesting and a great presentation. And then Matthew Kudek said, can you comment on approaches that you think may be successful in helping to increase the T-cell infiltration into neuroblastoma?
Yeah, it's a great question. There's a lot of work here. So if we're talking about infiltration of engineered T-cells, there is a lot of work, as you know, on armoring CARs, and that can be by co-expressing interleukins, co-expressing immune checkpoint inhibitors, trying to, there are TGF beta decoy co-receptors, so co-expression. So there's a lot that we're doing there. In the GPC2 example, the initial CAR that will go to the clinic will be a non-armored CAR. But what Chris will be working on as soon as we get the final all of the IND enabling work done is testing a variety of different armored CARs. In terms of getting normal host T cells into the tumor, we think that is why chemoimmunotherapy works. For many of you who take care of high-risk neuroblastoma patients know that a major recent breakthrough has been showing that dinutuximab, when combined with low-dose chemotherapy, is highly effective, response rates of at least 50% in refractory patients. And so we think that is the mechanism that the chemo is causing an inflammation, allowing NK cells and perhaps T cells to get into the tumor microenvironment. So that's not proven, but that's still a hypothesis. But yeah, a lot of people working on this, and we actually have at our next major moonshot meeting an entire session on how to make a cold tumor hot, and a lot of it is going to be exactly what you're asking.
Thanks, John. The next question comes from David Schulman. It says, thank you for a great talk. Can you talk more about how your strategy to target MHC-bound proteins differs from enhanced TCR therapies and the strategies to get around HLA class restriction?
Thanks, David. And I'll embarrass you by telling you I had a virtual happy hour with your dad the other night, that stepmom. So I think the vision, whether it's enhanced TCRs or CARs in these ultra rare conditions, is that we're not going to be able to get around the HLA class restriction unless we can develop enough of these to have a panel to cover the majority of HLA genotypes that we'll see in our population. So the lead CAR and TCR that we're working on is directed against FOX2B. I showed you one example, but we now have multiple different SCFEs. The same peptide is presented on some other HLA alleles, and other peptides are presented on other HLA alleles. So we're developing a warehouse sort of approach, so that, if you're A2, you get this CAR or TCR, and if you're A24, you get this one. And we think that, with the way that things are advancing and the throughput with which we can do these sort of experiments now with multiplex tetramers and that sort of thing, that we should be able to solve that problem, which has really hampered the field.
Well, thank you, John. That was the last question that has come in. So we appreciate it. We're coming up on the 1 hour, so it was perfect timing. And I just want to let everyone know that we have more lectures planned in the series. You can go to the website to look at the schedule. If you have ideas for more topics, for more talks, send me an email, j@alexslemonade.org, and we will look into getting those topics covered. But the next couple that we have scheduled, one is on pediatric brain cancers as epigenetic disease. We have one on zebrafish modeling in sarcomas. We have another one on Beckwith-Wiedemann syndrome, and we have a few others planned but aren't up on the site yet. But you can put your email address in on the site. We'll notify you when new ones are scheduled. And also, if you have suggestions, again, email me your suggestions and we'll try to get talks on those topics. Any closing remarks, John?
No, and I should have mentioned when I was asked the MIBG question that Alex has funded a REACH project for us where we're trying to make a next generation MABG, where we're substituting acetine in for iodine, which we think will be more potent. So shout out to Alex's for funding that work.
Will that do anything for the toxicity?
Yeah, it should be an outpatient therapy, and it should be much less toxic. It shouldn't cause any of the hematologic toxicity.
Excellent. Exciting. Well, thanks again, John. We appreciate it. And thank you for everybody for joining us. We got 100 people that stayed on till the end. So that's always a sign of a good talk.
Okay, thank you, everyone.
Bye, John.
Bye-bye.